Okay, this video is going to be the super simplified version of cellular respiration, making a map of the 3.5 stages involved in cellular respiration. Before we get started with actually making our map, I just want to point something out to you. And our starting material for cellular respiration is one molecule of glucose, which is made up of 6 different carbons. Okay, you can count the carbons. And is arranged in this ring-like structure. Okay, what we're going to do is we are going to focus on what happens to those carbons, just to kind of simplify this process. Again, this is probably the most complex process. I think it's the most complex process in biology. Okay, so we're going to work our way through and we're going to simplify it as much as we can by really just focusing on what is happening with the carbons. And to make it a little bit easier to visualize, I'm going to put the carbons in one straight line so that it just, I think, visually makes it easier to understand. But I do want you to keep in the back of your mind that, realistically, this is going to be in this ring-like structure. Okay, so keep that in the back of your head. The first step of cellular respiration is called glycolysis, which literally means the breaking down of glucose. There is 0 oxygen that is required for this reaction, so it can occur with or without oxygen, and it occurs in the cytoplasm of the cell. We are going to start then with our glucose. Okay, remember 6 carbons. There's 6 carbons here. 1, 2, 3, 4, 5, 6. I put them in a linear form. All right. What we are going to do, the end result of glycolysis, is we're going to break the 6 carbon compound up into 2 3-carbon compounds. Okay, and we're going to do this one time, break it up into 3 carbon compounds, and then we're going to kind of rearrange those carbons again to produce a different 3-carbon compound. So what we're going to do is we're going to use some energy. We're going to invest 2 molecules of ATP to break apart this carbon compound into 2 3-carbon molecules. And we're going to call these 3-carbon molecules right here PGAL. I should just put this here to not get super confusing. So PGAL is a 3-carbon compound. It's got these phosphates on it. The phosphates came from when we invested the ATP. The ATP broke apart into ADP and released a phosphate group. Those attached to these 2 3-carbon chains, and we end up with this chemical called PGAL. What we're going to do now is we're going to use those phosphates to now slam them together with ADP and make ATP. And we'll do that for each particular molecule. So we're going to 2 ADP on this one will form 2 ATP, and we're also going to have a reaction with the hydrogens here where hydrogens get added to NAD plus to form NADH. So if you didn't watch that super simplified video of ATP that is posted right above this, you really should, just to remind yourself of what these compounds are. But they're energy compounds. These are what give our cells energy. Okay? Each one of these 3-carbon PGALs are going to do that. So this one's going to produce 2 ATP, this one's going to produce 1 NADH, and this one's going to produce 2 ATP and an NADH. And the compound that's left over after this reaction is called pyruvate, or sometimes known as pyruvic acid. This is the end of the first step of cellular respiration. We started with the 6-carbon compound, glucose. We ended up with 2 3-carbon compounds called pyruvate. And in the process of converting this to this, we made 4 ATP total, 2 from this one, 2 from this one, and 2 NADHs. So we made 6 total energy storing compounds. Now, remember that we had to invest an initial 2 ATP, so our net gain is going to be a little bit different, just because we got to subtract those 2 out. We had to put some energy in order to make some energy. This process occurs in the cytoplasm of the cell, so in all that semi-fluid stuff that the organelles are floating around into. So at this point, If you have any questions about glycolysis, you should stop, write them down quick, shoot me an email, and let me know what they are, so that I can answer them. The next stage of cellular respiration is called the Krebs cycle, also known as the citric acid chain. Okay, this is a pretty complex cycle involving a lot of changes in chemical structures of different chemical compounds. But before we can get to glycolysis, we have this transition stage. So you can see I kind of drew this little line right here down so that you can kind of see where the stages end and where the stage begins. But this is a transition stage, which is basically kind of a prep stage for Krebs cycles. There's just this little thing, this little tweak of those pyruvates that need to be done in order for it to enter into the Krebs cycle. It's not really a part of glycolysis, it's not really part of Krebs, so we kind of call it this transition stage. And here's what happens in the transition stage. Now remember, we're just going to focus at this point on one of these pyruvates, okay? Let's just focus on this one pyruvate here. This 3-carbon pyruvate is going to undergo an oxidation reaction where a carbon dioxide will be lost. Okay, so we're going to lose a carbon on here. It's going to get oxidized and released as carbon dioxide. And in that process, we're going to make another molecule of NADH. So, in this process, we lose a carbon, and this new 2-carbon compound is called acetyl coenzyme A. That carbon is right here. So we're still accounting for all of the different carbons that we have. This acetyl coenzyme A is a chemical that we're going to use to enter into the Krebs cycle. Okay, 2-carbon compound. Now, that leads us to the Krebs cycle. The Krebs cycle is going to occur in the mitochondria of the cell. So here's a rough drawing of the mitochondria. You have your outer compartment here, you have your outer membrane, you have your cristae, you got the inner compartment or the matrix inside. So what's going to happen here is that this is going to occur in the matrix of the mitochondria. This whole reaction is going to take place in there. So what happens here in the Krebs cycle, oh, and another important note, you have to have oxygen. Oxygen is required for this step to occur. If it is not there, then we're going to go into a whole different pathway, which is fermentation. So we need to have oxygen for the Krebs cycle to occur. So let's go back to where we left off. We left off with this chemical called acetyl coenzyme A at the end of the transition stage. Acetyl coenzyme A is going to merge with a chemical, a 4-carbon chemical called oxaloacetic acid. So I just have it labeled here, oxaloAA acetic acid. It's going to join with this 4-carbon compound. So if we join 2 carbons with 4 carbons, we're going to end up with a 6-carbon compound called citric acid. This is why it's called the citric acid cycle. Citric acid is the same stuff that's found in your lemonade, it's found in your orange juice. Now, the citric acid cycle is called a cycle because basically what's going to happen here is this citric acid is going to cycle through a whole different varieties of chemical compounds until we end back up with this oxaloacetic acid. Now, for our purposes, I do not want you to know all of the different names of all of the different chemical carbon compounds that it's going to turn into. But I do want you to understand that we have what is a cycle here. Okay? All of these, there's going to be all of these rearrangements of the carbons until we get back up to this oxaloacetic acid. And in the process of rearranging all these carbons, we are going to produce some energy-forming compounds. We're going to produce an NADH here in this stage where this is a stage where they're going to rearrange themselves. And then we're going to produce another NADH here. We're going to release carbon dioxide along the way. 2 molecules of carbon dioxide. That's what happens with the 2. We had 6 here. We're going to get rid of 1 for carbon dioxide, get rid of another one for carbon dioxide. That's what leaves us with our 4 carbons. Okay. Later in the cycle, we're going to produce one full molecule of ATP. We're going to produce one molecule of FADH2, another molecule here of NADH. And then we're going to end up back with our oxaloacetic acid, which will merge with the next acetyl coenzyme A that comes in, and the cycle will just keep on going. And it'll keep on producing these different varieties. Okay? Occurs in the mitochondria, in the matrix of the mitochondria. So, who cares? Well, we formed one molecule of ATP. We know why that's important. That's like the ultimate energy guy in our cells, okay? But we formed a whole bunch of FADH2s and a whole bunch of NADHs. And so why this is important is because these chemicals are going to be the ones that are going to enter into the electron transport chain, which is the final stage of cellular respiration. Okay? ATP your cells can use right off the bat. But these need to get put into the electron transport chain in order to make the more important and stronger energy compounds ATP. So at this point in time, I want you to, I need to go back and stress something here. We focused on what happened with one pyruvate. Remember, I put this piece of paper over here and we looked at just this one pyruvate molecule going through this process. Well, you need to keep in mind that this pyruvate is also going through the exact same process. Okay, they're going to be doing it simultaneously. It's just easier for our purposes to focus on what happened to one, but remember, it's going to be also occurring up here with this 3-carbon pyruvate as well. It's going to form acetyl coenzyme A. It's going to start into its own Krebs cycle. So, at the end of the Krebs cycle, here's what we have. We have one cycle produces NADH, but remember we have to go through it twice because there are 2 pyruvates, so multiply that by 2. We produced, this was in the transition stage, we produced 3 NADHs in the actual Krebs cycle, so again multiply that by 2 because there were 2 pyruvates. That gives us 6 NADH for a total of 8. We produced 1 FADH2, 2 times, and we produced 1 ATP times 2, so 2. So at this point, after glycolysis, after the Krebs cycle, this is what we have total. 10 NADH, 4 ATP, and 2 FADH2s. ATP will get used directly by the cell. These guys, NADH, FADH2, they're going to enter into the next stage, which is called the electron transport stage. Now, again, I am going to make this as simple as possible, but please know in the back of your brain that this is an incredibly complex system. I am putting it in super, super simplified form. But here's what happens. Let's just take an NADH. It's going to leave the Krebs cycle, because it was produced over here, and it's going to enter into this electron transport chain, which also happens in the mitochondria, and it's going to happen between the membranes of the outer membranes and the cristae, the folds. So this NADH is going to go through an oxidation reaction, which means it's going to ditch off its hydrogen. Okay, here it is over here. Kind of like how when the ATP got rid of a phosphate, all right? That leaves us with NAD plus hydrogen, plus some electrons. This is going to be in a super high energy form. And what it's going to do is it's going to go through a series of reactions that are going to rearrange things to give us some different intermediate compounds. And, you know, these aren't, for our practical purposes, I don't need you to know these, but just understand that it's going to lose energy here, energy gets lost. It's going to convert this to this, energy is lost. It's going to convert this to essentially 2 electrons, 2 hydrogens, and some oxygen, which will rearrange to form water. And in this whole process that's occurring, let's move this over this way, you have this loss of energy. Well, the question is, why lose the energy. Where is that energy going? What is it that we're going to use it for? Well, what we're going to use it for is that lost energy is used to pump hydrogens that are sitting here inside. Here's your mitochondria. Okay, here's the matrix. You've got all these hydrogens in the matrix. That energy that's lost in the electron transport chain is going to pump these hydrogens across the membrane, across the cristae, these folds in the mitochondria, into the outer compartment here. Once these hydrogens start to gather out in this area, it's a process of simple diffusion. There are so many of them, they want to go back inside where there's more room, okay? And that's just a natural process. So the hydrogens get pumped out, then they just flow back in, and as they flow back in, they slam phosphates and ADP together in order to make ATP. Hydrogens pumped out, they flow back in. As they flow back in, they slam together ADP plus a phosphate and make ATP. Out, come in, make ATP. So for each NADH molecule, we will get 3 ATP that's made from this whole electron, which really you could call it the hydrogen transport chain. We'll get 3 ATP that's made from that. And the same general thing happens with FADH too. Okay, it's just a loss and gain of hydrogens, and for every FADH that enters into the electron transport chain, we will get 2 ATP that's made. So if you sum all of that up, we will end up getting 30 ATP from the NADH molecules that we made. Remember, if we go back to where we started, we had 10 here. We'll take 10 times 3, and we're going to end up with 30 ATP. We had 2 FADH2 molecules, so we'll take 2 times 2, and we're going to get 4 ATP from the FADH2 molecules. And then we made 4 normal ATP through glycolysis, where we made a total of 2, and the Krebs cycle, where we made a total of 2. So altogether, this process produces 38 molecules of ATP, the ultimate chemical energy source. Now, know that this isn't a perfect, ideal situation. Typically, our cells are not quite this efficient, and we will make less than 38 due to other environmental factors. But if in a perfect world, going through this entire cycle, we should end up with 38 ATP. Okay? So real quick summary with your map here. Start with glycolysis, start with glucose. At the end of glycolysis, you end up with pyruvate. Pyruvate will turn into acetyl coenzyme A, which will enter into the Krebs cycle, making citric acid, changing, and as all of those rearrangements occur, these things are produced as byproducts, okay? And eventually then these things, NADH, FADH2, are going to enter into the electron transport chain, where as they lose energy, they pump hydrogens out that flow back in, causing it's like a physical reaction of ATP to be formed. Ideally, you get 38 ATP that form. What I want you to be able to tell me about this process here, let's go back to here, is I want you to tell me the beginning and end results of each phase. Okay? And I want you to be able to look at this whole map that you made and be able to answer some questions like, what happens if we can't produce acetyl coenzyme A? What's going to happen to the amount of ATP that's produced? Well, what's going to happen if for whatever reason you have a disorder of the mitochondria where you don't have hydrogens that can come out, or whatever? I want you to be able to look at this whole map and think about what the consequences will be if certain things happen based on what you know. So I would highly recommend making this map. It's going to help you. Obviously, when you use it on a test, you can fold the whole thing out and kind of see here if I give it the long view. what it looks like. I used a legal sheet of paper, but you can easily do it on one. But it's basically just a map of cellular respiration. So I hope this gives you a nice simplified version of what I consider to be the most difficult topic in biology. And in 20 minutes, you just cranked through the most difficult topic of biology. So right on, you should walk proud and tall today. Have a great day.